Good afternoon and welcome to the Health and Beyond Research and Innovation Showcase, where we're going to see my group, the Limb Preservation and Wound Research Academic Unit, showcase where we're going to look at innovation in limb preservation. Firstly, I would like to acknowledge the traditional custodians of this land and pay my respects to the elders past, present and emerging, for they hold the memories, the traditions and the culture of the Aboriginal and Torres Strait Islander people across the nation. My name is Dr. Matthew Malone. I am the Director of Research for the South West Sydney Limb Preservation and Wound Research Academic Unit. It's an absolute pleasure to be here with you this afternoon. I would like to invite Professor Josephine Chow, who is the Deputy Director of Research and the Director of Strategy and Partnerships for the South Western Sydney Local Health District, to deliver our opening address.
Thank you, Matt. Yep, I would also like to acknowledge the traditional owners of the lands on which we are meeting today and pay my respect to the elders past and present. Unfortunately, Dr. Amanda Larkin, Chief Exec, and also my boss, she's not well today. So, but she actually would extend her welcome to all of you in the virtual seminar. I'm very delighted to see all of you and some more people tuning into across our community in Southwestern Sydney and local, national, and also international guests who will join in and jump into this exciting limb preservation and wound research in Southwestern Sydney and the collaboration nationally and internationally. I will support and what's been discussed in the past about the great work that Dr. Matthew Malone, the director for the academic unit, and also Professor Hugh Dixon, our favorite clinician, and also the board member for the LHD. Both of them will be actually spending some time and sharing their work in research to all of us. And all the distinguished guests joining in Southwestern Sydney, both local, national, and internationally as well. Looking at the program for today, for the seminar is outstanding already, without even listening to that yet, but it's actually the presentation, including lots of new technology in wound repair and regeneration, and then it's a lot of those innovative approach. Yes, my job is actually part of the T2 for the local health district, and I have been including into the health research and portfolio in Southwestern Sydney. And we actually got a research strategic partnership and research strategic plan that we've been using and focusing to make sure that we can actually use that to not just translate our research into the community to provide care, but also to actually deliver work such as recruiting the best workforce and retaining and nurturing emerging researcher and clinicians as well in all disciplines. So I'm very excited to see some of those pioneer research today, and then how the care and translation into tomorrow into the healthcare system. Some of you may know about the Health Beyond Research and Innovation Showcase. This is not the first time we run the showcase. We used to run before COVID period. We used to run in person, 2 days full seminar in June, face-to-face with close to 1,000 delegates from the local health district and the guest delegate as well. Unfortunately, last 2 years and this year, we have to run it differently. We adopt the new way of living that we actually been running it on a monthly seminar virtually, and has been very successful with quite a bit of attraction. And also I think the good thing is you can actually review those presentations afterwards as well. So it's actually the technology allow us to do that. The Health Beyond Research and Innovation Showcase, a partnership between the local health district, Southwestern Sydney, and also the Ingham Institute for Applied Medical Research and the university partners as well. And we actually using innovation and support that, and then try to including local and international speakers and the partnership into translating research into a bigger audience. Academic units is actually come to my heart. Back in 2015, when we actually appoint or recognize, acknowledge the first group of academic unit, today we got 10 group. From diabetics into the best of the limb preservation and wound research. We got child psychiatrists and women's health. Some of them actually will be showcasing end of the year as well. This academic units is not just created, but they are funded investment from the local health district, and then to build into the multidisciplinary team to conduct clinical trials, initiate research, and also ultimately revolutionize health outcomes. So the structure of them are really important, that we understand this is actually not just pop up overnight, and it's a very sensational vision from the district and from the chief exec as well. So thank you once again for joining today's seminar on this important research area. I particularly again thank you for the limb preservation team and the research team, academic research team, for hosting this seminar. And I hope you will be informed and inspired by the speakers in this critical research area, which will shape the care that we will deliver in Southwestern Sydney, and also will translate it to the rest of the world. And enjoy the presentation today.
Thank you very much, Josephine, for the introduction. And, I would like just to step back for a minute and acknowledge Josephine, Amanda Larkin, Professor Bouquet, and Southwestern Sydney Local Health District. What they've sort of done, and the vision in, as Josephine had pointed out, invest in academic units. And for context, our limb preservation group were awarded $2.25 million in 2018. We're coming into the last year now of our funding. And, I think most people who are involved in this area know that it's a rich area that is really not very well funded. It's underfunded. And so to have the acknowledgement and the investment from the health district to acknowledge that this is such an important area, not just locally, but globally, has been an amazing for our group. And I'd really like to thank them for their, I'd like to thank them for trusting us with their money, and in return, ourselves performing and undertaking high quality research so that we ultimately improve the lives of people in Southwestern Sydney community and broader. I could sit here probably all day and tell you, the costs of diabetes, foot disease, amputations. It'd tell you that it's a significant health burden and contributes to major health economics, the financial burden that's from both the health provider and the patient themselves. I could sit here and tell you about how diabetes and the 5-year mortality rate for people with diabetes-related foot disease is worse than most cancers. And I could continue this for at least an hour. But I thought that the best way to maybe address this is to hear from one of our patients and to summarize in George's own story what it's been like for him living with an amputation and a wound. And so this is my friend George, a patient of ours, been with us for many years, who has consented to having this film done and shown it as part of this showcase. And so I'd like to share with you over the next 5 minutes George's story.
Can you tell me, what is it like for you as a person living with a chronic wound?
I had the amputation 10 years ago in October. This year will be 10 years since the amputation. At times it's difficult. I was active in work-wise and family-wise and otherwise, and now not doing anything is difficult. Doing menial tasks takes longer. That hurts, like washing dishes, hanging up clothes. That was never ever an issue in my life. I just did it because it was there to be done. Work-wise, I was a storeman, and that means standing and walking. Then I went to being a courier. That meant a lot of driving, not just 8 hours a day, sometimes 12 hours a day, sometimes 16 hours a day. It depends where and how. I stopped that completely of work, which is difficult. At 50, I still had the desire, wanting to work. It's what I've done all my life. I've always worked. The hardest thing is to convince a government department that you can't work, and trying to get enough, whatever there is, qualified for NDIS, but the government says no, you can't get a disability thing. That has been very traumatic. It puts you in depression. You get depressed over the littlest things, a wife and children, and it's at times you get loneliness. But I'm lucky I have nieces and nephews that adore me, and that's one of the reasons why I can't do that S word. I'm not going to say the word. I don't use that word ever again. But I couldn't do it because of the ripple effect.
When you come into the high risk foot service,
Yes.
What are your expectations about the care you receive?
Number one, the care I'm receiving, there aren't any adjectives to say how highly and how much they have helped me getting used to not having my toes and being there and helping me in every shape and form. As for adjectives, the adjective I can think of is helpful, caring, wearing their hearts on their sleeve, being absolutely treating me with dignity and respect, which is the two most cornerstones of any kind of treatment. As my expectation, after 10 years, all I want is to heal. I feel like I'm an annoying person. I feel I try and make them laugh as much as I can because I can laugh then as well. The help and support has overwhelmed me, especially the support mechanism. I'm going to cry because I love them dearly. They don't realise how much they have helped me just by being someone to talk to, someone to understand my grievances, understand how difficult it is not to do the things I did before, and not being able to do those things anymore hurts. Not only physically, but mentally, and it's difficult at times. Some of the doctors that I've seen, there's one I want to strangle. He said, you can't.
And so, very, very powerful message from George, apart from the last bit about strangling doctors. I think that just really summarizes what our patients go through and the reason why we're here doing what we do. I want to thank George for allowing us to show the video and the time for that.
Can you tell?
Okay, so the program overview that we have today, this is what we're going to follow through. We're going to have a few live speakers who will come on and present live. And then we also have other speakers who unfortunately couldn't make it here today or are overseas, but have kindly provided us with their presentations in a recorded format that we'll go into. We will not have a sort of a traditional or formal Q&A. There is a Q&A function in Zoom. And so if anyone has any questions that they'd like to ask, then they should use the Q&A function, not the chat function. And what we'll do is at the end, we'll pull them all off and we will send them out to each speaker to answer directly. And hopefully this will be fine with everyone. But our first speaker, an absolute pleasure to have this lady with us today, who is one of the people I look up to, particularly with research that I'm doing myself. And it's a pleasure to introduce Professor Alison Cohen. And I will stop sharing my screen so that Alison can pull up her own talk.
Okay. Okay, so that's all good, is it? Yes, yeah, fantastic. So thank you, Matt, for giving me the opportunity to come and talk to you today. I've known Matt for quite a few years now, and it's very kind of him to invite me. So just a bit of background, I'm a professor of regenerative medicine and deputy director of the Future Industries Institute here at the University of South Australia in Adelaide. And I've been doing wound research now for over 25 years. I came to Australia in 1996 and have been leading a wound research group all that time. And I guess what I want to do today is just to give you a little bit of an overview, just very briefly in my 10 minutes, about some of the work that we're doing. So I can just move it on to the next slide, which doesn't seem to want to do. There we go. So, as you all know, we're dealing with wounds that just fail to heal. So, why are we requiring new technologies and new treatments? So there's currently, as you know, quite a lack of knowledge about how to diagnose and treat wounds. There's an absence of really good therapeutic agents that we can use to actively promote healing responses. There's a lack of diagnostic tools for identifying people who are at risk of wounds. And there's also a lack of predictive indicators to guide clinical practice. So these are some of the areas that my group has been working on for quite a few years now. And I guess I've sort of divided them into these 4 different areas. So we're keen to investigate and develop different therapeutics. We're developing advanced dressings. We're looking at different sensors and diagnostic approaches. And we're also interested in developing different technologies, and all aimed at all types of wounds. So yes, focusing on diabetic foot ulcers today, but also interested in burn injuries, in venous leg ulcers, in chronic wounds, in all the different types of wounds that are a problem on a day-to-day basis. So focusing first on our work on wound therapeutics, and I know that there's far too much information on this page, but I just wanted to introduce a protein that we've been working on now for the last 20 years or so, called flightless. And this is just a target protein that we've identified to be a really important key bioactive agent that is present in wounds that actually causes wounds to fail. And so because of the work we've done on this particular protein, we've been developing an antibody therapy that we can use to treat different wounds. And we do a lot of in vitro studies in looking at different cell types, and we do a lot of in vivo preclinical studies using a range of different animal models, such as acute wounds, burns, larger animal models such as pig wounds, and also diabetic wounds. So we have several different models of diabetic mouse wounds, such as a streptozotocin-induced model of impaired healing, and also a genetic DB mouse model. And when we have been treating our different types of wounds with an antibody that neutralizes the activity of flightless, then we're able to show that we get improved healing outcomes. And this is just summarized in some of these pictures here. So when the wounds are treated topically, so either with a cream or a gel containing the antibody, we're able to improve the rate and response of healing. So one area that we're now currently working on, and this is, while this is a burns focused, it's an NHMRC Ideas grant that I've currently got with Fiona Wood and Mark Fear over in Western Australia. We think there could also be applications for diabetic foot ulcers. And I guess the approach there is that to develop an immunotherapy that can actually promote healing responses when injected just into the body rather than topically onto a wound. Because we know that Phytolus impairs healing responses, and we're also aware that it's secreted into the environment of a wound. So we're keen to neutralize its activity. So one of the approaches that we've been taking is to actually intradermally inject this antibody therapy into mice. And then with either an excisional wound or a burn injury, we've been able to show that we can improve the rate of healing over 7 days compared to our antibody therapy versus just a saline or an irrelevant antibody control. And we're able to significantly improve the quality of healing. And also what we've been able to do is to label our antibodies that we inject and show and follow them and show that they actually do go straight to the site of the injury where they are able to mop up this protein that impairs healing responses. So this is an area that we're really interested in developing more and will be an area of focus because we do believe that having an antibody therapeutic is a straightforward approach for treating wounds. We're also specifically looking at diabetic wounds in a Department of Defence US funded project with Professor Rob Fittridge from the University of Adelaide and Nico Volko from Monash. And here what we're doing is incorporating our antibodies into porous silicon nanoparticles because we know that diabetic ulcers, diabetic foot ulcers have a really high proteolytic environment. So it's important that when you're developing or delivering a protein therapy into a wound that you protect it from those enzymes that are present within that scenario. So in this project, what we're doing is developing this way to deliver functional and active antibodies right to the surface of these diabetic wounds. And we've been able to show that if we encapsulate the antibodies into these porous silicon nanoparticles, we can significantly improve healing responses. And this is just in an acute wound, but we also did it in a diabetic mouse wound as well. And we were able to show that we could significantly improve the rate and the quality of healing. And we were also able to show that the nanoparticles were protecting the antibody cargo from proteases that were present in the wounds. So this is still quite early stages of this project. We're looking at moving this into larger animal models and looking at other types of particles, but we hope to be able to progress this into clinical trials in the near future. So as far one of the other areas that we're really interested in developing is advanced wound dressings, because we know that although there are multitudes of different types of dressings currently on the market, none of them are actually able to stimulate healing responses. And I guess because we know that there are a lot of dressings that just sort of cover the wound but don't really enable that wound to be able to heal in an active way. So we were part of a cell therapy manufacturing CRC, which enabled us to work with some really good biomaterial scientists, particularly Rob Short, who has now moved back to the University of Lancaster in the UK. But we're also able to work with Giles Kirby and Louise Smith, who were able to develop a surface that could be applied to a dressing, and it can be applied to any dressing at the moment. And we're specifically putting it onto a patch that we've developed, and that can actually then enable us to attach stem cells to that surface. And while we've been looking at stem cells, it can also be used to deliver other types of cells, such as keratinocytes or fibroblasts, that can then be used to stimulate that healing response. So we've developed this plasma polymerized surface, and this is plasma that is actually, it's not blood plasma, it's actually plasma such as you'd have in, it's cold plasma, it's like what you get in a TV, a plasma TV. And this plasma is able to functionalize the surface and then enable us to put different chemistries onto that allow the cells to attach and to adhere, but not too well that they don't come off, because we actually want to deliver these cells into a wound. And we've currently, we've tested these patches that we're developing, which we call SciPatch, in preclinical animal models, such as in mice and in pigs. And we've been able to show that they stimulate healing. And the results have been so interesting and so encouraging that we've been able to start a phase 1/2 clinical trial. And we're doing this with Rob Fittridge again at the QEH and the University of Adelaide, in combination with 2 industry collaborators, so TechSite, who are able to develop the surface for us, and then Cynata, who is a company that develops stem cells. So we've just started this randomized double-blind placebo-controlled phase 1/2 study. We got our ethics approval middle of last year, but it took us 6 months to be able to actually start the trial. And so far we've had 3 patients recruited into the study, and the results are looking really promising. And hopefully in about 6 months' time, we should be able to tell you a bit more about how well the trial is going. So this is actually the very first instance where I've been able to perform research that started out in a laboratory. We've taken through all the preclinical trials necessary, and this is the very first human clinical trial that we've been able to take all the way from bench to bedside. So it's something that we're really proud of and excited to see how it will progress in the future. So, and another dressing that we're also trying to develop at the moment is one that can actually in real time measure the level of infection and neovascularization of burns in the first instance, but we're also wanting to put this into diabetic wounds as well. So this is a collaboration between researchers at RMIT, led by Brant Gibson, who's a biomaterial expert, and also with some researchers and colleagues at the University of Adelaide who are experts in biophotonics. And then we're doing all the preclinical analysis of this new dressing. And the idea for this dressing is that we're able to incorporate nanodiamonds, which have a very temperature sensitive way of measuring the environment, and some pH sensitive fluorophores, because we know that when a wound is infected, you get an immediate increase in temperature and a change in pH. And if we can measure that early enough and at a, prior to the infection actually taking hold, then it will be possible to be able to treat a patient much earlier with antibiotics and potentially reduce the amount of systemic antibiotics that patients are given. So the idea is that this will be a dressing that you can see through it, and it will change colour in response to infection. And also, as part of this project, we're using optical coherence tomography to actually scan these dressings for the vascularization. So this is able to monitor whether or not the wounds are starting to revascularize, and which is an indicator of healing. So this project started this year, and we're making some really nice inroads, and hopefully again we'll be able to give you some more information about that in the future. So another area that we're really keen to work in and develop, and I just wanted to mention to you today, is the ability to develop different diagnostic platforms, not necessarily to monitor wound healing, although that's a really interesting approach, but we're also in preventative wound management, because we're keen to be able to support clinicians to be able to identify patients who are potentially at risk of developing a diabetic foot ulcer, even before that ulcer appears. And the reason why we think that this is a possibility is because we've been able to collect bloods from patients with diabetes before they get a wound. So we've been able to collect bloods from 48 months before patients, we've been able to track patients for 48 months and been able to sort of divide those into patients who didn't get a wound, a diabetic foot ulcer, and those that did acquire a diabetic foot ulcer. And then we looked at our flightless protein, which we know is a biomarker of impaired healing. We found that patients who went on to get a wound within 48 months had really elevated levels of this protein present in their bloods. So we think that potentially that flightless is a biomarker that could be measured in a thumbprick blood spot, potentially similarly to how it's measured when insulin levels are measured or blood glucose levels are measured. And it could be a way that clinicians can monitor a patient's potential risk of acquiring a wound and being able to implement different strategies to help them negate that risk. So we're working with some really clever analytical chemists led by Professor Emily Hilder, who has already developed a HemaPen, it's called, and this is a diagnostic agent, a device that can be used to take blood samples, and then it has a wicking system inside of it that you can kind of perform a Western blot within a pen. And we're able to, and what we're doing over the next few years, as we've just been funded for this research, is to actually make this a really quick and responsive blood test, similar to a rat test for biomarkers of impaired healing and able to predict wound risk. We've also working on diagnostics and sensors, again with Nico Volker this time again at Monash. And what Nico has been able to do and what we were able to do was to be able to measure the level of this protein again, which is my favorite protein, flightless, because we do know that again, it shows up impaired healing responses. So what Nico has been able to develop is a label-free electrochemical detection platform that can sensitively and rapidly detect the levels of flightless in wound fluid. And being able to do that can then also give an indication of whether or not a wound is likely to heal. So not only predict people at risk, but also predict people who are likely to have impaired healing responses, because we have been able to show that wounds that have high level of flightless, such as diabetic foot ulcers, do take longer to heal. So again, we are developing different strategies, different approaches to be able to monitor biomarkers that we can identify to be important within wounds, to be able to then use that as really important diagnostic tools to inform clinical practice. So the final one that I wanted to quickly mention was wound technologies. And here we're really interested in photoantibody therapies. So we're working on an approach that we can use photosensitizers, which we can link to bacterial proteins, so that we can measure when a wound becomes infected. We can use a sensitizer that binds directly to bacterial proteins present in a diabetic foot ulcer, for example, and then use a photosensitizer using near infrared light to activate ROS, which is present within the environment to kill those bacteria. So this is a really targeted approach. Photoantibody therapy is routinely used in many different areas, but it's quite a broad approach and can potentially kill healthy cells. So being able to target it more directly to bacteria, we would be able to kill those bacteria without harming any of the surrounding tissue. So that's another project that we're currently working on. So I guess what I just wanted to give you a feel for today was not any specific, I guess, outcomes of these studies, but just really an overview of the kind of approaches that we're taking. So, you know, against all those different areas. So, you know, with our antibody therapy, which is a therapeutic approach, I talked to you about the Cy-Patching, but we're also developing advanced silver nanoparticle dressings, and then our other approaches in diagnostics and technologies. And just to say that this is done by my group based at FEE and led by a lot of really, really talented research scientists who are all very keen to try to improve healing outcomes for patients with all types of wounds. So thank you, Matt. Yes.
Thank you, Alison. That was an amazing talk. Thank you very much. If I just get you to stop sharing your screen, and now I can reshare mine with this back onto you. So again, huge thanks to Professor Cohen for an amazing talk. I told you he's doing some really, really cool stuff. It's really exciting what we're going to see over the next couple of years come out of her lab. Thank you again, Alison. We're going to move on to someone closer to home who is located a few yards down from the clinic from me, which is Professor Hugh Dixon. And Professor Dixon is going to present his talk on something which I think is really exciting and very close to home to me. Again, Prof, I will stop sharing my screen so that you can share your screen.
Yep. A sec. We right to go? Okay, so this is a proof of concept study of Recce 327 topical therapy for mild skin and soft tissue diabetic foot infections. And a couple of questions immediately come up. Well, what's Recce 327? What does it do? And I'll sort of talk a little bit about that. I'm going to talk a bit about resistance to anti-infective agents. I'm going to talk a bit about biofilms and diabetic foot infections. That's very brief. And then give you an overview of the study which we've designed to test whether Recce 327 is effective as topical therapy in treating acute shallow ulcers of the feet in people with diabetes. So what is Recce 327? Well, it's an Australian developed anti-infective agent. It's synthetic, and it's entirely novel. Well, sort of novel. The two agents that make up Recce 327 have actually been around for a long time. It's 5% oligomer and 35% in polyethylene glycol 200. The 200 just refers to the molecular weight of the vehicle. So acrolein, which is one of the things which gets polymerized, is an interesting molecule, 3 carbon atoms, a hydroxyl group sitting on one end. It's highly reactive. It's actually the smell of burnt fat, which is intriguing. But in polymers, it forms most interesting substances. It's the base of a lot of plastics. It's also the base of a lot of gels that are used in wound care. For instance, Zetavit, which is probably known to a lot of the clinicians here as a superabsorbent wound dressing, is actually made up of an acrolein copolymer, and that's what makes these superabsorbent gels. Ethylene is also an extraordinarily simple organic hydrocarbon, or hydrocarbon, they're all organic. It's a simple alkene. It's the simplest of the alkenes, and again can polymerize. So for instance, in polyethylene glycol, we've got polymerization of the ethylene moiety with hydroxyl groups added on to each end. And polyethylene glycol is also found in wounds, in wound treatment. A Solugel, I think, is probably the best example of that. And it works essentially on wounds by promoting hydration and having an osmotic effect on bacteria. But when you combine the two of them, so on the left of your screen, you've got the acrolein molecule with the hydroxyl groups and the polyethylene glycol. What this does is disrupts the cell wall. It binds onto the cell wall. It's not simply an osmotic pressure effect. It disrupts the intracellular metabolic systems and the cells lyse. So it's bactericidal, the bactericidal component being dose-related. Interestingly, it's active against the major pathogens, the escape pathogens, which are Enterococcus faecium, Staphylococcus aureus, Klebsiella pneumonia, Acinetobacter baumannii, Pseudomonas aeruginosa, and Enterobacter species. Systemically, in animals, it's got a short half-life. It ranges between about 40 minutes to 2 hours. So it's cleared pretty rapidly from the system. It's not a microbial product. So at this stage, there's no developed microbial genetic resistance mechanism. And that's actually quite important because resistance can occur against all the anti-infective agents pretty well that we've developed, especially antibiotics, but also against biocides such as the biguanides or protosan, which is polyhexanide, against the phenols with triclosan or the polychlorinated phenoxyphenol, and also the quaternary ammonium compounds with substances like benzalkonium chloride and chlorhexidine, which we're all very familiar with. So bacteria and fungi have managed to develop mechanisms of resistance either by changing the cellular impermeability by modifying their cell wall structures, or by biofilm formation where they secrete slime that protects them from substances in the environment, such as antibiotics or biocides, by developing efflux pumps. So as a material moves into the cell through the membrane, it's pumped out again. Mutations at the target site of the agent, the anti-infective agent, so the target site actually modifies, so it's either incapable of being bound, or there's an overexpression of it, so many more targets are now present, and so the anti-infective agent is essentially mopped up before it can do any damage to the cell, or by producing specific enzymes which degrade the antibiotic. So far, because Recce hasn't been exposed to the bacterial flora of humans, we haven't got any resistance problems at the moment. But what we worry about is biofilm, which is a sort of really a tool. It's a man for all occasions. It's something that it's a slime that colony or colonies of surface-associated microbial cells enclose themselves in, and it prevents the agents from actually getting to the microbe. It's a major problem in diabetic foot infections, and it can lead to failure of both topical and systemic therapy because of this inhibition of penetration of the antimicrobial agents to the actual substance of the cells of the microbes. It's more of a problem in chronic wounds, and often the only way to overcome the problems associated with the biofilm is to actually remove the tissue containing the biofilm. So surgical debridement is required. Is there an optimal method of treatment of shallow ulcers in people with diabetes? Well, the answer is no, we don't know one at the moment. There are some general principles in the care, though, of people because they tend to be recurrent. We'd like to avoid exposure to systemic antibiotics wherever possible, because once we expose people to a systemic antibiotic, we induce growth of resistant species. We'd like to avoid exposure to topical antibiotics, so we won't give them systemic, we'll give them topically, but topically you've got the same problem. They still will induce resistance, plus they tend to be overrepresented in skin reactions. So dermatitis following applications with topical antibiotics is very frequently seen. So we tend to avoid agents in treating diabetic foot infections, agents such as mupirocin and neomycin and frammycin and chloramphenicol. Now, these are often very commonly used in ENT work. What about other topical agents? And there's a whole list of them. So we've got a whole lot of topical agents we use for diabetic foot infections, some of which have got an effect and some of which there's some evidence for them, but no strong evidence. Povidone iodine or betadine, gadexamar iodine, everyone's aware of, iodosorb, a whole variety of silver dressings which rely on the release of silver ions into the fluid surrounding the microbes in order to disrupt them. Polyhexanide or prontosan, honey, which works essentially by osmotic pressure on the cells, and methylene blue and potassium permanganate, which are strong oxidizing agents. So where does Recce 327 fit into this? Well, we know from the animal studies that it's got strong antimicrobial activity. We know from the toxicological studies that it's reasonably safe, though they do indicate that it's probably better given topically than intravenously. There's been a first in human study in a patient with burns in Perth where it was used as topical therapy, as spray on. And so what we've devised is a single centre prospective interventional study with a spray on anti-infective treatment with mild skin and soft tissue infections. It's a phase 1/2 study, so it's a very early study in terms of the drug development. It's essentially a safety and efficacy study, and we've got 2 dosing schedules we're going to use. The first cohort will have 16 people having a spray on Recce 327 daily over a 14-day study period to their shallow ulcer. And cohort 2, we're going to run it every 2 days over the 14-day period. The reason to go for the 2 days is because that more closely resembles what we do in actual clinical practice. People get into the study if they've got a mild wound infection, and we're going to use the wound infection score / foot ischemia score for inclusion criteria. Then we're going to leave people out who've got severe infections. We're going to leave people out who've got severe or moderate ischemia. They can't come in unless they're over the age of 18. They've got to have either 1 or 2 type diabetes, and they've got to be newly diagnosed, and they haven't been able to be used any topical antimicrobial or antibiotic therapy in the 3 days prior to enrolment. We're going to exclude people who've got severe, moderate to severe infections and moderate to severe ischemia, who've had multiple antibiotic regimens, who look like they've got osteomyelitis, if too small, so if less than 1 centimetre, we're not going to have them in. And of course, we're not going to take in people under the age of 18. And we're not going to take in people with documented severe peripheral arterial disease, pregnant women, people who are known to be allergic to 327 hasn't been around for long, but PEG 2200 has. And anyone else who we think, well, I don't think we'll put this person in. Because we're going to say, oh, I don't think we'll put this person in, we may also try to, in some way, alter the who gets what, either a daily or second daily treatment. So we're going to use block randomization to avoid selection bias. We're going to do tissue culture biopsies for microbial identification. We're going to use a standardized wound assessment using the diabetic foot injury score. We're going to use digital photography for monitoring progress. We're going to be blood monitoring for safety and inflammatory markers, and we're going to do a pharmacokinetics study to look at how much of the Recce 327 actually gets across the skin. Primary and secondary outcomes, of course, we're interested in the change in wound size, which we think we should be able to manage pretty readily with our photography systems. We've published extensively in that area. We're going to be examining skin reactions, plus change in the microbial counts on our cultures, the need for rescue therapy. That's to say, when people have got a bailout, we think, oh, that's not working, we better introduce antibiotic therapy. We're going to be looking at changes in laboratory tests, and we're going to be trying to see whether the comorbidities that people have any influence at all on the outcome, though the numbers are small. We're going to see them pretty regularly. I could say day 0, we'll try and get them in, and then they come on day 3, day 7, day 10, 14, 21 and 8 and 28. So the main thing to notice, it's a 14-day study. So at 14 days, we stop and we evaluate, see what the effect has been. Where are we up to? Well, we've got the method for spray application. We've got the tools for assessment, and everyone's been trained in them. They're used routinely within the department anyway, so that's no big deal. We've got a population of patients who are ready for recruitment, and we have submitted ethics application for our protocol, and we're waiting clearance. So watch this space as developments occur.
Excellent. Thank you, Prof. This is one I'm really excited about. We've been involved in the study, and we've been fortunate enough to obtain a grant for this from the company to do this trial. And I think it'd be really, really exciting. Again, you know, anything, the possibility of good local wound care and local delivery to reduce, you know, maybe the overuse or unnecessary use of systemic antibiotics to reduce the burden of antimicrobial resistance. So moving on from Prof's talk, we have Dr. Tabby Ahmed. She is a staff specialist in vascular surgery with myself and Prof Dixon at Liverpool Hospital. She came over from the UK a couple of years ago now, and has really enhanced, you know, the way in which we deliver care with regards to vascular surgery, and just really becoming an integral team member. And Tabby is also doing another project with us, and Tabby is not here, she's in the theatres, so she has kindly recorded her video.
Hello everyone, and thank you to Matthew for inviting me along today. I'm so sorry I can't be with you in person. As you can see, this is a pre-recorded presentation, but that's because I'm currently upstairs in theatres operating. Over the next few minutes, I'm going to spend some time telling you all about an exciting new randomised controlled trial where we're going to try and determine the causative agent and optimal duration of antibiotic therapy in persons with diabetes and foot osteomyelitis. Now that title's a little bit of a mouthful, and so it's been shortened to the bone biopsy trial, or the BBOP trial. And this really represents an exciting new international collaboration between us here at Liverpool and Campbelltown Hospitals, and Edgar Peters and his research team from the Amsterdam University Medical Centres in the Netherlands. And it really has come about as a result of this abstract that was published late last year that we read and recognised to be an important research question, particularly in our own patient population. So we got in touch with Peter and we asked. The aims of the study are to try and compare the outcomes in those patients with osteomyelitis who were treated with antibiotics targeted either to their bone biopsy results versus the deep tissue or ulcer bed biopsy results. The working hypothesis behind this is that we think that treating patients on the basis of the actual infective pathogens within the bone will demonstrate superior treatment outcomes and healing outcomes compared to those who are treated on deep tissue samples. So why is any of this important? Well, it's important because the diagnosis of osteomyelitis can be really challenging, and trying to figure out which antimicrobials to use and which pathogens are actually causing the infection is also challenging. The reference standard from all the international and national guidelines for diagnosis is a positive bone culture, either obtained surgically or through a percutaneous bone biopsy. But these biopsies are actually quite difficult to obtain, and in part it's a combination of a number of factors. For surgical biopsies, well, not everyone has an operating theatre where biopsies can be performed, and that's particularly true in rural settings. There's also a perception that undertaking a bone biopsy is cumbersome, time-consuming and expensive to perform. But this has to kind of be put into context of current practice and our own patient population. So at present, if we want a bone biopsy, we have to get in touch with our radiology colleagues. We then have to liaise with them for them to undertake a bone biopsy percutaneously. And southwestern Sydney has, one of the highest rates of diabetes in all of New South Wales. Cumulatively, our high-risk foot services see in excess of 150 patients a year with diabetic foot ulcers, of whom 40% are complicated by osteomyelitis. And from work that we've performed within our own patient group, we know that bone biopsy results compared to which we think are the actual infective pathogens driving the bone infection have a low concordance with superficial wound swabs and deep tissue cultures. The bugs that you grow in both samples are actually quite different, and the concordance is as low as 22%. And treatment for these patients, particularly if you're just using broad spectrum, antibiotics, is already lengthy, it's already costly, and it often results in a digital amputation, if not a major limb loss. And that may in part be because we aren't actually treating the osteomyelitis with the right antibiotics targeted to the actual infective pathogens within the bone. And in fact, the whole issue of taking bone biopsies is currently a little bit of a hot topic. In last year's Australian Diabetic Foot Disease Guidelines, one of the future research considerations that they postulated for osteomyelitis was centred around taking bone biopsies and how perhaps we aren't doing them often enough. So this study really fits in quite nicely with one of the current Diabetes Feet Australia research interests. So the study design, it's a two-armed, non-blinded randomised controlled trial. As part of this, we're going to analyse the metadata of those patients presenting to our services with diabetic foot ulcers and suspected diabetic foot osteomyelitis, and we're going to compare the two groups, which are those who are treated on the basis of bone biopsy results versus those who are treated on the basis of deep tissue culture results. The planned numbers for recruitment from southwestern Sydney are 91 patients, and this is going to be matched by our colleagues in Amsterdam. And the primary outcomes that we're looking for is ulcer healing and remission of their osteomyelitis at 12 months. And secondary outcomes include the time taken to remission, evaluation of a series of lab results performed in recruited patients at set time points in their journey through the trial, comparison of the actual results that we get from the two sampling techniques, and we're also going to undertake some quality of life surveys of patients to see what their experience of living with osteomyelitis is like. So where are we at? Well, as with all studies or trials, the first stumbling block is to actually get your paperwork together and get it through the ethics committee. So we've gone through Regis, and I'm pleased to say that that is all now approved. The second stumbling block, particularly when you're trying to bring a new technique into the hospital, is to source, locate, find money for and purchase the kit. So I'm pleased to say that that is all also sorted, and we now have bone biopsy kits in the hospital. So all in all, we're pretty much good to go. So what happens next? Well, we need to start recruiting, and hopefully that's going to take place from next month. So watch this space, and we will be back in touch once we've got some results. Thank you very much.
I just want to thank Tabby for her talk. She was a little bit quiet, so apologies. I was getting a few pings about the volume. We'll try and address that maybe in the video recording and see if we can turn it up from our end. That study is going to take place in the foot clinic at Liverpool Hospital again, and I think again is going to be another a great resource in trying to improve care by making sure we're actually targeting the correct microorganisms when we're delivering antibiotic therapy for osteomyelitis. So huge thanks, Tabby, for taking the time to do that presentation before you ran off to theatres. The next up we have is Dr. John Frew. I'm really interested in this talk, mostly because we part funded some of John's work with a small grant from the academic unit. He really is quite an intelligent guy, and the dermatology research he's doing is pretty cool and slightly close to my own heart with regards to using genomic platforms. And so let's jump in and hear John's talk.
Good afternoon. My name is Dr. John Frew. I'm a dermatologist and researcher based at the Department of Dermatology at Liverpool Hospital, as well as the head of the Laboratory of Translational Cutaneous Medicine here at the Ingham Institute. I'd like to speak to you today about some of our preliminary work, which has been supported by the Southwestern Sydney Limb Preservation Small Grants Scheme regarding inflammatory circuits in pyoderma gangrenosum. So first of all, I'll go through a few things in this short presentation today. First of all, what is pyoderma gangrenosum? What do we currently understand about the pathogenesis of PG, as we call it? And a little bit about our translational research pathway in terms of identifying acknowledged and well-documented inflammatory pathways in this disease and identifying whether they have clinical relevance in terms of novel therapeutics and novel outcomes in the condition. So this is what we typically see with pyoderma gangrenosum. It's a incredibly painful, highly burdensome disorder. Although it's relatively rare, 1 in 100,000 people per year, it certainly has an increased prevalence amongst those with coexisting hematological malignancy, inflammatory conditions such as hidradenitis suppurativa, inflammatory bowel disease, rheumatoid arthritis, and the chronic relapsing inflammation that's systemic in these participants are thought to predispose to this aberration in wound healing, leading to these rapidly deteriorating chronic ulcerations, both on the lower limbs as seen here and also in peristomal sites, which are quite burdensome for these patients and very recalcitrant to all sorts of standard therapies, including high-dose immunosuppressants. So novel therapies are desperately needed, and it's really being held back by our lack of understanding of the pathogenesis of the disease. So the pathogenesis is incompletely defined. Interestingly, the name is quite misleading. Pyoderma suggests an infection, and this is not an infectious process. And certainly there is no gangrene, there is no deeper extension down and infection in deeper structures which lead to loss of limbs. Certainly it can be misdiagnosed as an infection. And traditionally in many textbooks, as well as in teaching, it's thought to be a primarily neutrophilic process. But we certainly do see, and we'll speak about this later in our work, that mixed inflammation, mixed innate and adaptive immunity seems to be very prominent in this condition, which brings it away from what's traditionally taught. And certainly the multiple inflammatory associations that we spoke about previously are documented here as well. So this is from one of our systematic reviews, identifying the different aspects of innate and adaptive immunity that can contribute to PG. Certainly it's thought to be a dysregulation of innate immunity surrounding follicular structures, contributions including pro-neutrophilic drivers such as CXCL1 and CXCL8. We know that dendritic cells and T cells are strongly involved, but certainly we do see other innate immune markers such as IL-15 with polarization of macrophages and natural killer cells that do contribute as well. Later on, with florid destruction of the surrounding tissues, we do see activation of fibroblast markers and a very mixed inflammatory infiltrate, but there's very little known about what happens during this scarring phase and if there are other mixed forms of inflammation which are seen, as it's really covered over by a very dense neutrophilic destructive infiltrate, which has really hidden away a lot of the main pathogenic players in the disease. And certainly we do see loss of all adnexal structures, loss of follicular units, leading to inflammatory resolution eventually, and a lack of recurrence at previously involved sites. So one of the main pathways which has been implicated is that of the Th17 immunity or type 3 immunity pathway. And this is thought to be largely driven by that innate function surrounding hair follicles. We know that Th17 pathways are strongly implicated around here, and CXCL1 and IL-8 are pro-neutrophilic drivers, drawing neutrophils into the dermis and through the epidermis to help promote inflammation. And that inflammation is thought to lead to dysregulation of wound healing and epithelial mesenchymal transition pathways that lead to this chronic rapid ulceration. Certainly we know that complement C5AR1 particularly is known to very strongly self-amplify neutrophil signals, and this has also been identified as prominent in PG as well. So some of our initial work with many clinical patients who present with PG has definitely shown that there are dense inflammatory infiltrates, as we see here in some basic H&E, but certainly these are relatively mixed. We do see strong neutrophilic signs, but we also do see a lot of dendritic cells, particularly when there starts to be dense periods of edema surrounding the superficial dermal infiltrates, and definitely clusters of B cells and CD20 positive pre-B cells and plasma cells, the markers with CD138, that tend to cluster around vessels. And this certainly implies that there's a bit of a mixed immune function here where possibly adaptive immunity may help co-opt neutrophilic infiltrates so that the immunological pathways involved may seem a little bit more complex than they're initially thought when we first examined this disease. And certainly interactions between B cells and neutrophils have been identified in other diseases, and we're seeing this as a possible link to more complex immune dysregulation in this disease in patients with PG. So the premise of our translational work is that if this is solely a simple neutrophilic disorder, then IL-23 blockade, which is the link between dendritic cell activation, T cell activation, particularly Th17 activation, and pro-neutrophilic drivers will be enough to stop this inflammatory infiltrate and hopefully reduce inflammation and result in a clinically significant effect. And what we aim to see is a downregulation of CXCL1 and CXCL8. But certainly from the initial clinical work that we have, we don't seem to see a clinically significant response over a 12-week period with very high dose IL-23 antagonism with PG. Now, this is something that is really opening the door to a deeper understanding of what kind of pathogenic mechanisms are involved here, because if the pre-existing paradigm is true, IL-23 should stop this Th17 polarization and then reduce this inflammatory infiltrate and allow for self-resolution through the pre-existing population of IL-10 producing Tregs present in the dermis, but this is not occurring. So this is something which was quite unexpected. And the next steps in our work now is to say, well, the Th17 axis is not seeming to be a primary player in this disease, and then what is? So the interpretations could be that there is modulation of the pathway, but not enough to produce a clinical effect, or certainly that the Th17 axis is more of a bystander and there are more complex mechanisms at play driving this complex neutrophilic infiltrate into the dermis in PG and leading to the lack of epithelial integrity. And certainly we don't have a large amount of understanding in terms of the interactions of adaptive immunity and epithelial integrity, specifically in skin. So our next part of our work is to then take a lot of the tissue samples and serum samples that have been taken through this work, analyze them both for biomarkers of disease severity and clinical response, and see, once we take away that IL-23 mediated Th17 axis, what is left and what correlates well with disease activity. And certainly we anticipate that it may be a Th17 independent complement-based pathway, or certainly from pre-existing data, is there a stronger role for these populations of CD20 cells that we see clustered around vasculature deep in the dermis of very active PG tissue? And so this is something that will hopefully open up novel pathways of pathogenesis and identification of novel therapeutic targets for this disease, which could certainly change the overall paradigm of treatment for patients with this recalcitrant chronic burdensome disease. So I'd like to thank Dr. Akshay Flora, my master's student who is primarily driving this research, as well as Matthew Malone and Hugh Dixon as co-supervisors of his work. Certainly we've had great support from the Ingham, as well as the small grant scheme, as well as the university and Sun Pharmaceuticals for provision of the medication itself. So thank you very much, and we hope to be able to update you very soon regarding further biomarker and mechanistic work in this field. Thank you.
Thank you, John. And yeah, look, I think I'm again really close to home with this one. Really excited to see, over the next couple of years now what's going to come out from the research. He's been doing some great stuff in a quite a condition which has a significant effect for the patients that actually are attached to these wounds. Next up, it's my pleasure, absolute pleasure, to introduce Prof. Akinji. I say that with a rather overtone because we have some really exciting news that Prof. Akinji is going to jump in now and sort of just fill in everyone with what's been going on in the diabetes research space. So Prof, if you're around and you can jump on.
Yes, thanks, Matt. Can you hear me okay?
I can. I will mute myself so you can have the stage, and just tell me when you want me to change your slide.
Okay, I will. Thanks, everyone. My name is Elif, and I'm an academic endocrinologist at the University of Melbourne, and I'm also head of diabetes at Austin Health. And I have the pleasure of leading the Australian Centre for Accelerating Diabetes Innovation, or ACADI, which was formed really with the funding secured from MTP Connect last year. So it's a real once in a lifetime chance for us to help transform the field of diabetes and its complications. And we're really grateful to address the challenge for Indigenous and non-Indigenous Australians by delivering innovation on behalf of ACADI. And I'm really pleased to state that Matt Malone, Alison Cohen and Rob Fittrich are the leads of diabetic foot ulcers and neuropathy for ACADI. And this is a $10 million investment from the federal government administered via MTP Connect, and with co-funding of both cash and in kind of approximately 13 million, so bringing up to $23 million of investment for the next 4 years to help drive innovation in diabetic foot ulcers and neuropathy, diabetic kidney disease, as well as the acute complications of diabetes, including hypoglycemia and DKA. So next slide, please, Matt. So really our vision is to benefit people from diabetes at each stage of its diagnosis and complications, and just the previous one, sorry, and to deliver novel interventions. And this is possible because of the funding as well as our collaboration. So really it's a unique opportunity to unite Australia. Currently diabetes research is fragmented in Australia, and this collaborative virtual centre will bring researchers across the country, as well as engineers, data scientists, people living with diabetes and its complications, and scientists, podiatrists, allied health experts, and really being a bit of a template for innovation centres to help turbocharge innovation. And our values are excellence, integrity, equity, and innovation. And what we have with the CARDI is we have input from local and international experts. We have also CARDI platforms, including Biostats platform, health economics platform, behavioural platform, and co-design platform, and implementation platform to help support our 18 innovative projects, to help accelerate research and development in some of the most difficult complications of diabetes, including, as Matt eloquently discussed with his patient earlier on today, diabetic foot ulcers and amputations and neuropathy. And, you know, when I was preparing for this grant application, it just was staggering, the health economic costs of these complications. And everyone here knows about all the, as well as the health economic burden, but the impact that it has on quality of life. So such an important area of research, and we're really hoping with the CARDI to drive innovation in this space. So we'll have 18 innovative projects, and we'll also have a strong and national training program to help support PhD students and postdocs. So there will be opportunity with Western Sydney University being one of the core partners for people in the audience to apply for these postdoc fellowships, as well as PhD stipends. So, you know, what we want to be able to do is increase with these, get these products into the hands of people who really need it most, because there is actually a 19-year life expectancy for people who are living with these serious complications compared to people with diabetes who are not living with them. And just an example of this health economic saving is like even a 1% reduction in these burdensome complications will lead to 50 million per annum cost savings. So what we want to do is that over time, by uniting the country and allowing these acceleration of innovations, such as the innovative work that's been displayed here, and I'm really pleased to say that Matt, Alison, Paul Breen and Dinesh are going to talk about some of their really innovative work, which are actually part of the CARDI project portfolio. So what we want to be able to do is that, so over time, that when we go and do ward rounds in the hospital or do remote outreach service, that we really have a genuine reduction of these complications. So next slide, please. So with the CARDI, we have 70 organisations across the country, and these include many academic partners, 15 core partners and universities, as well as industry and health advocacy groups, health services, and commercial partners as well, who've come together to help work improve outcomes for people living with diabetes. And how we came about to the project portfolio is, you know, 70 across the country, our colleagues submitted 75 projects, which were then shortlisted for our research portfolio. And we really use the criteria of clinical impact and commercial feasibility as well, with the end user in mind. And so because of the 4-year timeline of the funding from MTP Connect, we didn't shortlist very early stage projects. And I guess the national network will really also result in an ability to undertake collaborative research, including clinical trials, and will help potentially aid with the development of an Australasian clinical trial network to help innovations across the country to progress to commercialisation, ultimately helping our patients. So, you know, I think the CARDI will really bring an opportunity for people across the country to collaborate. And next slide, please. So the collaborative gain is actually much more than the sum of its parts. So with the platforms and with our specialist advisory groups, both national and international, this will help pave the way to allow people to have access to these innovative products. And so, and we also recognise the importance of culturally sensitive approaches to research involving Indigenous people, and we'll use consolidated criteria for strengthening the reporting of health research involving Indigenous people. And we have a Indigenous advisory group led by Ray Kelly, who will also be reviewing the projects as well. And our governance is really designed to support transparency and portfolio tracking and effective decision making with input from expert advice. Next slide, please. So our short list of projects really are impactful and synergistic, and they're at different stages of technology readiness level progression, and they'll be supported by these platforms, and as well as also the training program of PhDs and early career researchers. And Aboriginal and Torres Strait Islander people with diabetes can participate in any of these projects, but the flags indicate more specifically studies which are highly relevant for Aboriginal and Torres Strait Islander people and will be conducted following community consultation. So rather than talking about each of the projects, in fact, you'll hear about the foot projects in detail. I'd just like to move on to the next slide, which is just to say that we have a very strong training program to help empower emerging and existing healthcare professionals, and also to enable collaboration with industry and with entrepreneurship skills for the next generation. And it's really important to note that the next generation we really need to support, because there is, I mean, especially with a massive clinical workload in diabetes with its complications, and then also with the COVID burnt, you know, a lot of people, clinicians are getting burnt out, and we really need to sort of nurture them and support them so they continue to have interest in research and continue with their research projects. So I think that with that, I would like to thank you, and would really like to hope to interact with you, and you could actually move on to the next slide, Matt. And this is really a huge team effort with investigators across the country, and it's really great to that Matt is leading this symposium today, and I'm really impressed by the quality of the work here, and I'm really great that we can collaborate with him, and it's fantastic to see the leadership in action. So thank you.
Thank you, Alif. And again, huge congrats to Alif and to Akedi on this fantastic win for diabetes. And so the next three presentations themselves will be on the projects which are going to be funded by the Akedi, and one of them is mine. And so I will jump straight into that. So I've been doing a lot of work with the group around the genomics-based approaches to basically try to look at wound healing and the biological or the cellular mechanisms and the microbial mechanisms related to foot ulcers and foot infections. And this study is basically trying to identify candidate biomarkers or biological and cellular processes that we're able to target. And what we really want to do, and you know, Alison set the scene beautifully for this. It's a real complex area, and I don't think we still have a true understanding of everything that's occurring within the wounds. Why do some wounds heal and some wounds don't? Why do some wounds go on to develop infection and others don't? What are the underlying dysfunctional host cellular or biological mechanisms which may explain why wounds don't heal? What are some of the microbial mechanisms which may lead to patients developing infection? You know, can we identify candidate biomarkers or cellular biological pathways that we can target to accelerate new therapeutics? And the same, can we identify microbial markers for new antimicrobials in a very similar way to what Alison has been already doing for quite some time now with things like the flightless protein. So the purpose of the study which we have is to better elucidate the impact of the social and intrinsic determinants on underlying molecular mechanisms which may influence health outcomes in individuals with diabetes foot ulcers. And what we want to do is identify the major influences on host immune, biological pathways and microbial factors, which probably modulate the immune and cellular dysfunction, which may explain why some wounds don't heal. Maybe look at the host microbe interactions, and together, how do they predispose individuals to the development of non-healing wounds or infection? And what we want to do is identify, again, similar to Alison, things like flightless protein, using a genomic approach, we want to identify biomarkers that will provide potential avenues for the development of targeted therapeutics aimed at maybe modifying the host or the wound environment, or producing anti-infective agents to combat wound microorganisms, and even maybe looking at biomarkers that are prognostic tools that can be implemented at the bedside as a point of care. What have we currently got now? We have a study which has already got ethics approval at Liverpool Hospital as the main site, and we'll be bringing online the Royal Adelaide Hospital with Rob Fittridge. We have to limit the study to 50 patients, and mostly because we're following those patients through their wound journey over 1 year. And what we will do at set time points is collect lots of biospecimens. We'll collect tissue, peripheral blood, and wound fluid. We'll also collect lots of clinical data and stuff around social factors. So we'll look at social isolation, loneliness, depression, anxiety, medication adherence, cognitive impairment, drug addiction. We're going to look at a raft of social factors as well to plug into our genomic data set. In addition to the set timelines, we'll also have sentinel points. So if a patient maybe develops an infection, we'll take an additional sample. If they resolve from the infection, we'll take another sample. If the wound gets bigger, we'll take another sample. And at each of those time points, we'll also track the progress of the wound. Is it getting better? Is it healing? Is it not healing, etc.? So we can plug those into our genomic data set. We're going to use an approach called RNA sequencing, or total RNA sequencing. It's going to produce huge amounts of data, over 24 billion genes, probably generating 5, 10 to 50 terabytes of information. And with that information, we'll have to use sophisticated bioinformatic approaches and models to analyze things around differentially expressed genes. So with the RNA, what we're actually looking at is the relative activity. So DNA holds our information and our RNA delivers that message to the cell for that instruction to be carried out. So when we talk about RNA sequencing, we're talking about relative activity. So we're going to look at the relative activity of host cells or biological pathways, and we'll also do the same for the microbial aspect. And our aim is then to identify some candidate biomarkers or pathways which we can pass over to Alison that she can then use in a similar way which you saw the flightless protein, so that we can hopefully in the long term develop effective therapeutics or diagnostic point of cares, for example, or even prognostic markers such as flightless in the ability to maybe detect a wound that might not do well very early on. What do we hope to find with our information? And so the easiest way is to maybe show you some of our pre-work, which we've already done in this area. We've spent quite a bit of time investing in making sure we can do the workflows, obtain the tissue, extract the DNA, walk it over 5 minutes to the lab, where we handle the DNA or the RNA, we extract it, we can sequence it, we can get actual information from the samples, and we can then use bioinformatic approaches to analyze those data and learn a little bit around how to best approach the bioinformatics. And so here's a sort of a scenario of what we hope to have from our data. We can see that we have a non-healing, but it's not clinically infected, but it's not healing. We treat him and we follow him, and the wound gets smaller and is healing, and we're taking samples at all these time points. The patient disappears for a little bit and comes back a few weeks later, and he suddenly got a little bit worse with his ulcer and developed a mild skin soft tissue infection. He's commenced on antibiotics and we treat him. But lo and behold, a week after, he comes back with an even worsening ulcer and has now developed signs of severe infection. And so we have time points through all of this sort of journey. And what we want to do is sort of look at the gene expression and biological pathways which are occurring through these time points. Let's see if I can just move this out of the way. There we go. And so some of the data which we've already looked at to give you a little bit of a sense here is looking at some of the microbial data, and we can just look at, and this is relative activity. So not just what's there, but who's actually being active with regards to their expression of genes. And we can see that we have streptomyces, some streptococcus, and some staphylococcus and other things in here during the sort of baseline visit to the healing visit to then coming to the infected visit. And we can see with a mild infection, we get quite a different jump in the relative activity of both streptomyces and streptococcus. And then when the person comes back with a more severe infection, the activity changes again and becomes a lot more diverse, mostly with some more fastidious anaerobes. So we can start to look at the types of microorganisms which may contribute to specific infections. And more importantly, actually come to look at the types of genes those microorganisms are producing within those different infections. So the mild to moderate skin infection and the more severe infections. And so these are maybe some of the markers which we may be able to identify in larger data sets that we're able to home in on and say, well, obviously, if we see this antitoxin here, this is predominantly not been noted in a more severe infection. These are maybe where we can do maybe dipsticks that can identify as a point of care some of these particular virulence mechanisms to differentiate the severity of infection, which will lead us to change maybe the way in which we treat these patients. Again, we can do these sort of multi-maps, which allow us to look at all of the cases we have and just look for patterns to see which ones are the most important, which ones trigger with severe infections. Is it consistent across the non-infected or the mild to moderate infections, to start to build a picture of what should be our target or candidate potential microbial markers, and even backtracking and relating some of these virulence mechanisms or proteins to specific microorganisms. So should we be targeting specific things? And coming on to more of the host measures, and this was from the gentleman which you saw, and we have a collection of these data now. And we can see that as the person moves through non-infected to being a mild infection to a severe infection, we can start to see different changes in host mechanisms which come into play. In particular, this SERPIN B2, which is actually a plasminogen activator inhibitor 2, which has been identified to have a role in the innate immune system. Again, this one of interest for us, PTX3 or long pentraxin 3, it's a part of the CRP superfamily, and it's switched off virtually in all non-infected, and even in mild infections it's off, but then it switches on for severe infections. And in the literature, there's already data which has looked at PTX3 as a potential biomarker to identify patients with sepsis and actually look at outcomes related to sepsis with PTX3 expression levels. Some other things, this FPR2, which is a pattern recognition receptor to bacteria, whether this would be something which we could look at. And again, here in the mild and severe infections is CXCL8, the precursor to interleukin-8 inflammatory chemokine, and whether this would be, maybe the next CRP or the next ESR, whether this would be beneficial. So this just gives you a look at the type of data we can pull. And here on the right-hand side, we can actually then use gene ontology to look at the different biological pathways which are either upregulated or downregulated, and again, may allow us to sort of target specific pathways we should be looking at more closely, taken forward into different animal models or confirming in our larger data set. So hopefully that just gives you a little bit of an insight into some of the data we hope to find and that we can use. Here's another one in a patient which we published on, a patient with a healing wound, and we looked at patients with healing versus non-healing wounds. And the two most upregulated or differentially expressed genes in patients who healed versus non-healing were identified to be as conical markers involved in B-cell activity. So, is this something which we start to focus on having a bit of a closer look? Is the role of B-cell activity in improving wound healing? And hopefully, our large data set is going to identify some of these things. A huge thanks to a very large team that it takes to do this, particular Dr. Tim Peters, who's a bioinformatics research officer with the Immunogenomics Lab at the Garvan Institute, my postdoc fellow, Dr. Michael Radzita, Associate Professor Slade Jensen, part of the WSU and our team, all of the high-risk foot service team at Liverpool tuning in, who help capture all of these samples for me and follow patients. Professor Alison Cohen for her great work in this project and taking what we find forward, and Rob Fittridge, who's going to be a site of ours, and also Dr. Stephen Kidd, who's going to be doing some work with our samples. As a whirlwind tour of what we're going to be getting up to with some of our funds, and we'll move on to Paul Breen, who is going to present his work, which is going to be funded by Acadie.
Hello, my name is Paul Breen, and I'm going to talk to you today about subperceptible stochastic resonance electrical neurostimulation, or SENS for short, a SENS device to improve symptoms of peripheral neuropathy in people with diabetes. Sorry I couldn't be with you there today. I wish I was. But let's start with what is SENS. SENS is, like I said, it's subperceptible. So in this intervention, it can't be felt. So the intention is that it is below the level at which someone will perceive it. It uses stochastic resonance, which is essentially using noise to improve function. My next slide will talk about that. And it is applied via electrical neurostimulation. So the intervention is applied via electrodes to influence nerve activity. So let's start with a little theory. Well, what is stochastic resonance? And essentially what it means is that you may have an input that it must reach a threshold. Here the threshold is a dashed line. And if the threshold isn't met, it doesn't cause any activation. So in this case, like a sensory receptor, if you push on the sensory receptor, it won't fire unless you meet some threshold. However, we can add in some stochastic noise, so some random noise that itself doesn't reach the dashed line threshold that we have here. But if you sum the two together, if both are applied simultaneously, you do meet the threshold and you do get the activity. So you can add in some level of noise that you don't perceive, but it has the benefits in terms of when a previously subthreshold stimulus is applied, you can now perceive that stimulus. Of course, this also implies that a very low level of stochastic noise will have no effect because it isn't enough to get above the threshold. But also, if the noise was too strong or the amplitude is too great, then it would be creating abhorrent action potentials in this case when you don't want them. So that's stochastic resonance, and that's the principle we're employing. There's also another really useful piece of information, and that comes from Arturo Rosenbluth. Interestingly, a very influential person, and great publications. This publication in particular, though, it never has only ever been cited 37 times, strangely enough. But it's a very interesting piece of work. In this, they use a peroneal nerve that they split in two towards the distal end. And they applied a supermaximal stimulus here on one of the branches. And they made a recording on the other branch. And sometimes they added a DC stimulus to the trunk. So you would generate your action potential. Action potential would travel up along the nerve, and you would see if anything gets recorded over here at the recorded electrode. So in the absence of this DC stimulus, as in case A, there's nothing recorded when the supermaximal stimulus is applied. That's what you would expect. But when they apply a DC stimulus to the trunk, as in case B, you do get a recording on the other side of the split. If you reverse the electrodes, you still get activity on the adjacent side of the split of the peroneal nerve. And then if you move this DC stimulus closer to the recording electrodes, you affect its latency, which pretty much proves that it is this DC stimulus that is causing this activity to appear in the recording electrode. Kind of bizarre. We are led to believe that axons, myelinated axons, these are myelinated axons, are immune from each other, but they do have apoptic interactions. They do have interactions between each other. So we've, and I'm just going to show you some of our own work. We've looked at compound motor action potentials. We stimulate at the elbow, we apply our SENS stimulus at the wrist, and we record the muscle in the hand, moves the finger. And what we find is that we do this over and over and over and over again. We have a minimum effect on things like the compound motor action potential peak amplitude. It's a little bit of a mixed bag in terms of what happens with various parameters around the compound motor action potential. But what is very reliable is its effect on the reliability of the compound motor action potentials. And what we see is a much narrower distribution around a lot of the metrics we look at. So here we see a broader, in the control conditions, we see much broader distribution of, in this case, the compound motor action potential peak voltage or peak amplitude. And that gets narrowed when we apply our SENS. And we see it's a rather consistent, not perfectly, but rather consistent response across different CMAP parameters, so amplitude, pulse width, and peak latency. We've also done this work where we've applied SENS to a finger while vibrating the fingertip and recording using microelectrode needles from the corresponding nerve. So the mechanical stimulus to the fingertip would look something like this. And then we are getting, in this case, unitary responses. So these are unitary action potentials for each vibration cycle. And what we find is we can compare this with psychometric evaluation. So what is the person, if we test their perception at their fingertip in a separate study, we find that they perform best with around 40 to 60% of what at the level that they would perceive SENS. So it's quite sub-perceptible, around 40 to 60% is maximal. And that corresponds to about 31 to 47 microamps RMS of current applied. And we see there is this inverted U-shaped curve of the response. So a little doesn't do much and too much is not useful. But also what we see around the same levels of optimal SENS being applied, we see a substantive reduction in jitter. So we can see essentially an increase in reliability of the interspike intervals. So let's put this together and think about what this all means, well, in theory. So SENS is close to DC stimulus. It's very close to DC, and it allows myelinated axons to be activated by impulses on adjacent axons. That's a running theory. This increases the number of axons fired for a given stimulus. So it's a form of spatial activation. Apoptically coupled axons influence each other, reducing action potential speed, but leading to greater synchronization. And it's only the last few years we're really seeing very good evidence that that is the case. Thus, spatial activation leads to greater apoptic coupling and even greater synchronization, seen as narrow distributions, as we saw in the compound motor action potentials, and reduced jitter in the single neurons. Is it useful? Well, we've done quite a number of experiments to look at the functional benefits. We see improvements in vibration perception in younger, older, and patients with diabetes. We also see improvements in ankle proprioception. And in this case, it's applied at the ankle and improves in an equal amount perception across the foot. So we can test the multiple sites, and the plantar aspect of the foot, we see improvements in each site. to the level of about 10 to 15%. We also see similar improvements in controls and controlled veterans, veterans with diabetes, and veterans with diabetes and peripheral neuropathy. In fact, if we look at the most severe cases, the ones that are actually, it says max 50, but we couldn't measure how severe their neuropathy was. They were higher than what we could measure, and they all come back within a measurable range. We also tested or had an experiment in Cape Town, in the townships in Cape Town, where we looked at HIV and non-HIV individuals and saw that we could improve their vibration perception as well in both HIV and non-HIV groups. How do we deliver it? Well, we have a pretty unique arbitrary waveform constant current stimulator, which is suitable for the purpose. It's not perfect, but it is probably the best stimulator of its kind in the world. And it has an associated app. You can create whatever stimulus you require, and it can do a whole bunch of stuff. So we have the technology that we're building upon to deliver the stimulus in a convenient manner. So why is this important? Well, foot ulcers are a precursor to non-traumatic lower extremity amputations in 85% of cases. A limb is lost to diabetes every 30 seconds. The peripheral neuropathy in diabetes is responsible for 27% of all direct care costs. This device, which we call a feeling aid to enhance sensory perception, could restore that protective sensation in 28 to 40%, we estimate, of patients with peripheral neuropathy due to diabetes. And that in itself could lead to a 40% reduction in the development of foot ulcers. Just a thanks to our partners and our sponsors, in particular the Australian Centre for Accelerating Diabetes Innovations, for this work, and thanks, Matt, for the opportunity to speak today. Thank you.
So thank you.
Hello, my name is.
Thank you for that, Paul. If you struggle to follow the biomedical engineering slides on that one, I think if nothing else, the last slide is probably is gold. And you know, if we can do what it says it can do and achieve it in that percentage of patients, we'll be on to a winner. Our next talk, and sorry, we are running slightly behind, is Professor Dinesh Kumar, who's going to talk on one of the projects, the Last Foot project under the Acadie banner. So I will stop sharing my screen and it will be over to Dinesh.
Thank you, Matt. Thank you, Matthew, for having me, and thank you. I think it's good work started off by Alif. Congratulations, and I'm glad to be part of the team who would be able to deliver some interesting stuff. Now, this is what I'm reporting now is a pre-published paper, and hence you will find that there are certain details which I am purposefully missing out. And this is, I happen to be the leader of the Biosignals Lab at the School of Engineering at RMIT University. And we've been working with our partners on thermal imaging and other imaging from modalities to detect the wound healing trajectory for the past nearly about 5 years. So this is a journey happening. Now, let me see if I can figure out how to.
Just if you click slideshow at the top, that should take
right.
you.
So this is a lot of information which perhaps for people in this audience may not be very relevant to you. Perhaps know these numbers better than I do. That a large population on the earth suffer from diabetic-associated foot or leg ulcers. And monitoring the healing progress and the progression of this is extremely important, because otherwise it can not only lead to into amputation and other such problems for the patients, but also it's a very expensive thing for the hospitals and the carers. There have been certain methods which have been proposed to be able to do the early detection. And so one of them is the two-dimensional and three-dimensional imaging modalities. The other one is hyperspectral imaging, and the third one and the last one is the thermal imaging, which have been proposed for this purpose by various groups around the world. So one of the problems with the RGB in imaging in the two-dimensional is that it leads to very poor sensitivity. To improve that, three-dimensional method were proposed. However, that does not seem to overcome the shortcomings either. Hyperspectral imaging has been proposed. There are two major groups that have proposed it. One is a group out of University LA, and the other one is out of Denmark. And both of them have got some nice results. However, on closer examination, it appears that you can get good results when in the lab, because there are certain there are lots of factors which contribute towards the outcomes, such as skin texture, skin color, ambient light conditions, and also it is a very expensive process, and it's also very slow. So the amount of data is large. The process in itself can be slow. So it is not something which when I once was speaking about this with Elif, and she said she wanted something which is handheld and less than $1,000. Well, this is definitely nowhere close to that. We're talking about a couple of $100,000 in this case. And then there is this conventional thermal imaging, which was found that while it seemed to have some positive aspects, however, it was very, very sensitive to the ambient conditions of the temperature, like, for example, the air conditioning, etc. And again, it was only suitable when the patient came in the third visit. So it wasn't something which you would get the answer straight away, which was, again, something which would not satisfy the clinicians. So an introduction to how does the image look like. So what you see is diabetic foot ulcer, where you got some information about the temperature of the epi wound center and the peripheral region, and you can also identify. So this image in itself, I was told by some clinicians, can be useful to them. However, this requires your own interpretation, and which may not be very accurate sometimes. So we're also having a look at the venous leg ulcers and looking at what happens with these images. So what you see on the top left are the raw images. We then have to normalize it. The whole intent is that we want to try and remove the effect of the background, which is going to have different temperatures, and especially if it's in a setting which is a non-air conditioned clinic. Then the intent is to remove the leg part from the rest of the image, and now to be able to then go back and refill the information about the thermal properties of the leg into the leg alone without the background. So this is a stage we refer to as a pre-processing stage, and it can be done to any thermal imaging, any part of the body, the feet or the legs or any other for that matter. The next, what I'm reporting is a small study that we did with the 47 unhealed and 17 healing ulcers. Now, we did both thermal and RGB images. We also did hyperspectral, though I would not be mentioning about that. We then, rather than worry about doing the conventional methods that people have been doing of trying to see how the images move across with time, we determined 19 textural features. And this was based upon some of the discussions we had with your colleagues, who said that they look at the roughness of the image of the wound to determine where it's going. So we decided that, and the whole data thus became 64 images, and each having 19 features. Now, we then decided that this was a large amount of data for the purpose of in the AI machine, and hence we had to do some reduction. For that purpose, we used the principal component analysis, or PCA, on these particular features. Having done that, we then divided the data into 70%, which corresponded to the training data, which is required for any AI machine, and 30% for doing the validation and testing. And we then repeated this 20 times in order to ensure that we have now covered the entire database and there is no bias in our analysis. Now, you would notice, unfortunately, I'm using the word feature 1, feature 2, and not giving the details because the paper is still under review and I would be in trouble if I gave more details along. Now, so we found that there were 3 major features that were most relevant in order to be able to distinguish between the healing and the unhealed wounds or ulcers. So what you see here on the left, on the first diagram on the left, is that you got the unhealed, which have got a higher value, the feature 1, compared to the healed. However, there is still a little bit of an overlap. So if you were to use it by itself, you would get a significant amount of error. Similarly, we also did for feature 2 the same thing, and you can see that there is again a large variation in the data. But the p-values seem to indicate that there is a significant difference between the 2 groups. Again, we did the same thing for feature 3, and similarly, we were able to identify a difference. P-value seems to be quite decent. In the end, then we also started to do the PCA to identify the most suitable, not just feature, but feature combinations. And when we did that, we got some very good looking AUC curves. So that thus indicates that it is possible to detect the wounds that may not heal in time. That is, in week 12, weeks was what we defined as a healing trajectory time. And we could do that right in the first visit of the patient. So right on the day 1, rather than have any repeats, the clinician would know if this person's wound is going to heal within 12 weeks or it requires extra intervention. So that, we think, is something which could be extremely useful and for the clinics, as well as. Then the next option we did was, we also started having a look at some of the RGB images. And we considered that typically the RGB images are being used only to detect the area of the wound, using which you have been able to, 1, keep a record of the images, and 2nd, you can also identify whether the wounds are healing by measuring the change happening over the first 4 weeks. But there is a problem. I'm sorry, I think in one place I've got the spellings wrong. It's healing, not hailing. So we thought, why don't we look at the shape of the wound? When we did that, so taking an example, this is the wound of and so we developed a software for this purpose to be able to do the automatic analysis. And what we found was that if we were to do the shape analysis rather than the area analysis, the results were significantly better. So what you can see here, for example, is that I've got the shape of the ulcer, which I'm measuring using fractal dimension, and that is giving me a picture. And this is a healing ulcer. So the one which is not healing, and even though the area may not be looking, may actually be looking as if it was reducing, but if the FD is not changing like this, then you could expect there to be a problem. However, this work is still in progress, and we do not yet have a definite answer on the use of this. A brief our team members, and this is our group who are, in this particular photograph, actually actively looking at one of the new imaging modalities which has just been introduced. I think that's my last slide, and some of our other partners. Some of the fundings that we've got towards this work have come from ARC, and defense, as well as a CRC. Thank you very much for listening. And any questions, or is that later?
Thank you, Prof. Kumar. Yeah, people are going to type questions as they go. So if anything comes to you, we'll direct it to you. I know you have a meeting that you have to jump off to. It's a great talk. And again, I think, the 3 projects are really exciting as to hopefully, having an endpoint which can translate into improving, wound care, diabetic foot care that we see in our patients. I will just ask you, Prof. Kumar, to stop the screen sharing so I can jump back on.
Thank you very much for having me on this.
Thank you, Prof. We appreciate you just hanging around. We are running slightly over time.
So I was enjoying your previous representations, including yours, with the hope that maybe with your excellent data, we could throw in some AI onto it.
Oh, I think so. I think there's some really exciting stuff that we can do with that. So on to our next speaker. This probably, hopefully, I would think needs absolutely no introduction to many of us. He really is one of the foremost leaders in diabetic foot research for, probably the past 20 years now. Professor Lavery is a fantastic surgeon, extremely busy. Whenever I ring him, he always picks up his phone when he's in theatre. He's an amazing clinical academic, producing over 200, 300 peer-reviewed manuscripts. His talents are enormous, but they don't seem to extend to technology. And I was hassling Professor Lavery for his presentation late last night, and he managed to send it across, albeit it is...
My name is Lawrence Lavery. I'm a professor in the Department of Plastic
him
Surgery
talking without a video. So I will leave you to Prof. Lavery.
Today I'm going to talk about hyperspectral imaging in wound healing. I really want to briefly address 3 topics for this presentation. First, I want to talk about the rationale for hyperspectral imaging. Second, I want to talk about hyperspectral imaging to predict ulcer healing. And then I want to finish up talking about hyperspectral imaging in surgical flap survival prediction or intervention. At the bottom left of this screen is an old hyperspectral imaging device that was commercialized to the product on the right by a company called Hypermed. That company is no longer in business. However, there are several companies in the United States that are commercializing this type of technology. What are the standard tools we have currently to use? So I've listed a few things that are commercially available and widely used in the United States. The most common thing that we have available in the hospital and in clinics are to look at arterial Doppler studies. So that allows us to evaluate systolic pressures of the dorsal pedis and posterior tibial artery and compare that to the systolic brachial artery pressures and look at those as a ratio. We can also look at toe pressures and toe brachial indices as another tool. In addition, we can look at patients' waveforms. Another tool that's commonly available in wound centers, but not generally available in hospitals in the United States, is transcutaneous oxygen measurements. What I really want these tools for is to look at amputation level selection to help inform me of tissue that is going to survive or not survive after intervention or when people are admitted to the hospital with infection, and technology that could help us understand wound healing potential in patients, perhaps to identify people that need more aggressive intervention or more aggressive wound therapies. one of the barriers that we have using traditional arterial Dopplers is that there's a really high rate of calcification of arteries in the lower extremity, which make systolic pressures and waveforms unreliable in people with diabetes. Ankle break. Brachial indices and toe brachial indices help us calculate macro circulation, and this information can be complementary to the data that we get from hyperspectral imaging. Transcutaneous oxygen monitoring information measures microcirculation. And I think the biggest limitation in using this technology is you can only evaluate tissue on the dorsum of the foot. So if you're trying to understand perfusion on the sole of the foot, you don't get reliable data. It also is pretty time consuming, and the new technology that is being commercialized by a couple different sources generally have a platform that is point and click with almost instantaneous evaluation of data that you can then assess and look at localized continuous variables to understand the local perfusion. Hyperspectral imaging can provide information on both the oxygen delivery and the oxygen extraction when we're assessing patients. Light-based perfusion assessment technology has really focused on using near-infrared light therapy to and this measures light reflected by hemoglobin and determines the proportion of oxygenated hemoglobin in the cutaneous tissue. Near-infrared light can penetrate the skin more deeply than visible light can. Near-infrared light has reduced light attenuation by melanin, so patients that have dark skin often don't get good information or a good signal. It's one of the limitations of this technology. Near-infrared light penetrates about 2 millimeters deep, and therefore it gives information about oxygenation in the upper portion of the dermis. So first I'm going to talk about the study that was published in Diabetes Care in 2007 that used hyperspectral imaging, and they didn't use this old technology. The next study has a similar format to the study that we just discussed. This was also a study that was using the HyperMed technology. So this was a prospective cohort study of 54 patients with 73 diabetic foot ulcers that were evaluated for 24 weeks. 54 of the ulcers healed and 19 of the ulcers did not heal during the evaluation period. The sensitivity, specificity, positive predictive values were also very good for such a small study. If you look at these graphs, the top graph shows a diagonal solid line that represents the decision line for the healing algorithm using oxy and deoxy hemoglobin values. If you look at the bottom graph, this shows the probability of healing based on the hyperspectral healing index or index for wounds that healed and wounds that did not heal during the 24-week evaluation. So using this bottom graph and ulcer with a positive healing index had a significantly higher likelihood to heal. So there are also studies that contradict these findings. So this is a study by William Jeffcoat that was published in 2015. It also was a prospective cohort study to look at using hyperspectral imaging to predict wound healing. So this was a study of 43 patients with diabetic foot ulcers, 26 healed in 12 weeks, 28 healed in 28 weeks. And what William Jeffcoat found was there was a negative association with tissue oxygenation and healing by 12 weeks. And there was a positive correlation between oxygenation assessed by hyperspectral imaging and the time to healing. This was a study from our group that looked at 40 patients that have diabetic foot wounds. And we evaluated oxyhemoglobin, deoxyhemoglobin, and oxygen saturation on the dorsal surface of the foot and the plantar aspect of the foot. And we found that there was no difference between healers and non-healers using these parameters. So not only did we not find some of the excellent results of sensitivity, specificity, and predictive value reported in other studies, we didn't find any association with these parameters and wound healing. You can see the images at the top show a patient's wound on the distal part of their transmetatarsal amputation stump minus a remaining fifth toe. So this is on the sole of the foot. You can see their oxyhemoglobin, their deoxyhemoglobin, and their oxygen saturation pictures, and the kind of the ordinal color scale on the left will give an idea of how this tissue at the periphery of these ulcers looked. So if you look at this data, we actually saw a negative association with dorsal oxygenated hemoglobin in that patients that had higher levels were non-healers. We also saw that patients that had higher levels of plantar oxygenated hemoglobin were also associated with people that were non-healers, although this was not statistically significant. But I thought a very interesting trend for such a small sample size. So I think there still is work to be done to understand this. I think we need larger studies. The thing that isn't controlled in most of these studies are all the other wound healing parameters that contribute to a high rate of healing, like aggressive offloading in either a total contact cast or a really good offloading boot product, the frequency of debridement, and all the other patient characteristics and comorbidities that affect wound healing. I mean, all of these studies essentially are assessing this measurement of perfusion as if it's the only important thing to evaluate in diabetic foot ulcer healings. And I think certainly treatment has a huge impact. So we really don't know a lot about the other treatment parameters in many of these studies. I'm going to finish by talking about hyperspectral imaging and surgical flap survival. So my hope when I started looking at this technology, especially in the new format that is essentially kind of a point and click device where you get almost instantaneous measurements of perfusion to the foot. And my hope was that this could be a tool that we could use in the operating room to decide the level of amputation or local flap survival. So this is just an example of the various measures of oxygen perfusion using hyperspectral imaging in a gentleman that has a rubber band placed around his finger. So you can see his oxygen saturation. The numbers are much lower than the rest of the hand. When you look at deoxyhemoglobin, this is in the blue range rather than kind of the yellow-orange range. Probably the most dramatic is when you look at the oxygenated hemoglobin, and this is, very low and much higher in the remainder of the fingers. Now, with this technology, for the instrument used in this study, you can take your cursor and point it over areas and get some continuous data. So this isn't just looking at a color chart. The color chart gives you an idea of where things are in relation to the other color spectrum, but you can get quantitative data with this technology. It's just not shown in this. Next, I want to show some examples using hyperspectral imaging to evaluate flaps in this rodent model. So this is called the reverse McFarland rodent model, and it's an example of using a flap on the back of a rodent. So the flap is designed so the distal portion can be ischemic. So you can see the changes clinically in this flap that has the distal portion is ischemic at 12 hours, 24 hours, 48 hours, and 72 hours. So we'll use this flap technique to show the value of hyperspectral imaging to identify injury and death to the distal portion of the flap before you can see this change clinically. So this is an example of the flap. It's a local flap that's marked on the back of this shaved animal. The pedicle is then raised on the sacral vessels. The wound is then surgically closed, and the flap is repositioned and held in place with sutures. So this is an example of a flap that has very little damage to the flap. So you can see the animal before any surgical procedure is done in the far left. Immediately post-op, you can see the flap with not very much in the way of ischemic color changes. After 2 minutes post-op, there are more ischemic changes, and this progresses in 5 minutes, 30 minutes, and at 72 hours, you can see that there is some necrosis just at the distal portion of the flap. So this is in contrast to a flap that you can see from the above slides, that it becomes, the distal 1/3 of the flap is very ischemic. So the pre-surgical pictures are very similar. There's a high oxygen saturation throughout the whole flap. Immediately post-op, you can see that there are a couple areas that are not well perfused, or the hyperspectral image is more ischemic. After 5 minutes, it is dramatically lower in oxygen saturation. The same thing at 30 minutes, and then eventually at 72 hours, there's full thickness necrosis of the distal portion of the flap. I'm going to end by giving a clinical example of a patient where we used hyperspectral imaging. So this is a 73-year-old gentleman who has had diabetes for 16 years. His glycated hemoglobin is 11%. He has a long history of tobacco use. and he presented with an infected ulcer of his fifth metatarsal head with abscess and osteomyelitis. So he had fourth and fifth array amputations and presents with this ischemic looking wound. So if you look at his angiogram on the left, he had 2 vessel runoff to the foot. So after this, the first surgery for source control, he was taken to the vascular surgery suite and was vascularized. So you can see that after revascularization, he now has 3 vessel runoff to the foot. His Doppler signals were stronger. The dorsum of his foot was warm, and he had a transmetatarsal amputation, and the medial skin that was still viable under his first, second, and third metatarsals was used to flap laterally and cover the soft tissue defect that was caused after the fourth and fifth array amputation. So he bled well. His soft tissue was warm and pink, and we were really optimistic after his really good revascularization that he would heal and have a good residual stump. So this is his pictures the next day after surgery. So, you know, if you look at the surgical site, you know, the values on both the plantar and dorsal areas of his flap are low, and that certainly reflects the discoloration and eventual necrosis of this flap. So in conclusion, I think functional perfusion, especially to inform this clinical decision-making, is still a big unmet need in diabetic foot complications. I think preliminary studies have really suggested that hyperspectral imaging may be a tool that's going to be useful to help us predict ulcer healing, and perhaps help us with surgical decision making for people that have localized flaps or free flaps to salvage their limb. I think there's still a lot of work to do for us to understand this technology. There's several companies that are probably all using a little bit different algorithms and a little bit different parts of the light spectrum to make their technology work. So we continue to do some work in this area, and hopefully it will be useful for physicians and surgeons in the future. Thank you.
So a big thank you there to Prof. Lavery. I'm going to move swiftly on because we'll just run in a tad behind now. And what better way to finish than with David Armstrong, a very colourful presenter, and again, one of the leaders in this field. A sort of slight declaration in that I'd heard him present this talk at a conference and asked him to do the same one, and he kindly just shared the video with me from that conference he talked at. So we don't have to reinvent the wheel, and I will just play David's talk from this other conference. So just don't mind too much the intro.
Armstrong is a professor who's tenured at University of Southern California. So from sunny Cleveland to sunny California, thank you. He has been everywhere and he has published so much. He even has a fun called program. It's salsa, whether it's the dance or the sauce, I don't know. I think he will let us know. I saw him sneak a cup of coffee before this, so I think he's ready to go. All right. Thank you.
How do you know if
Dr.
that was
Armstrong.
caffeinated or decaffeinated? There,
Oh, there
Professor?
you go.
We do not know this, and but it was carbohydrate rich. That's Professor McDonald. Anyway, listen, let's hustle. I'll be quick. I promise to get us in for some rich discussion. Let's make this happen. I put this slide up. I just put it up this morning after rounding virtually at our Rancho Los Amigos facility. This is these are iron lungs, you know, from the last great pandemic of polio, 1953, I think, and you see this led to modern hospitals. We're talking about hospitals and modern ICUs, which then made us prepared for this current pandemic as much as we could be. And then what has that done for us? I think maybe what it's doing is it's preparing us for what's next, which might be getting ourselves out of the hospital. So I think that's really what I'm talking about. I think it couldn't have been set up enough better than really to talk about that. I'm just a foot doctor, kind of a toe mechanic. My goal here is to kind of eliminate preventable amputation over the next generation. And I think with me and a lot of my colleagues in a lot of different disciplines, we're moving in that direction. We give away all our slides. I would just ask you to email me, ping me, copy them, make them better, pay it forward. We'll be talking about a bunch of this stuff in a very, very short scattershot amount of time. But we're really going to be talking not about hospitalization, although a little bit we will be. It's really about hospital-free days, if you will. Again, as we discussed, from this, at that same time, we see that this was going on. This is the very first kind of surgery ever caught on video. This is from Professor von Bergmann. I actually gave a lecture at his institution in Potsdam back in the day, but this is 120 years ago when he's doing this. And this is her first surgery. You see that amputation. And what kind of stinks is that this woman or this man having his or her leg off now 120 years ago probably had a better outlook on life than someone right now, somewhere around the United States, having her or his leg cut off. So I think there's a lot we can do in this area within diabetes to affect change. And it's really going to be looking back as well as kind of looking forward. You look at these gorgeous data, not from our unit, but from Amadou and coworkers just a couple of years ago in France. And what they were showing is even as in some places, you can see a leveling out of amputation, even though it's increasing in most places. And in France, it was leveling out at the beginning of the last decade. There was still an increase in incidence and prevalence overall background of diabetic foot ulcers, of wounds. So these wounds now are increasing even as amputations are leveling off, hopefully. So this is no different than cancer maybe 30 years ago, where you saw cancer deaths starting to level off, but just more cancer. So the denominator keeps increasing, and the rate of us having to deal with these patients it keeps increasing because we're dealing with acute on chronic complications, which is the bane of our collective existence. If you just want to try to put numbers to this whole thing, I mean, why not? This is from a technical review we had in the New England Journal not too long ago. And we said, you know, every second now someone develops a diabetic foot ulcer. About half of these get infected. About a fifth of those end up in the hospital. And of those, about a fifth get an amputation, which is why there's an amputation every 20 seconds now around the world. This is increasing in most parts of the world, still, unfortunately, not decreasing. It's we don't have time to talk about this in any depth, but we're going to talk about how to get these people out of the hospital, back to home, and back to living their life through various means of assessment, measuring, and managing. Because sometimes the best surgery is the one we never did. And we'd like to think that time's up, right? And but to that extent, let's just talk about these kind of foot ulcers and some of the things that are happening at our unit, at other units around the world that are looking to measure and manage tissue repair and wound healing and the patient in diabetic foot remission a little bit better. I think as we look at moving these patients out of hospital, we have to look at these centers, these diabetic foot centers, these wound healing centers without walls. And the pandemic has really forced this onto us in a lot of ways when we couldn't see all of our patients. And we see that we're moving now. And again, I'm preaching to the converted on this very, very fancy pants, non-Luddite call. But the big idea here is we want to move from these hospitals to highly capable non-hospital centers. And sometimes that's the home.
And it's becoming the home even more as we can measure more of what we manage, as many of the ICU technologies are becoming available in the home. And so I think this is super exciting. It's an exciting time to be doing this, even as a humble toe doctor. So let's, and what's really cool now is we have these sorts of programs that are going on. I'm the PI of our of this really cool National Science Foundation Industry University Collaborative Research Center at our place, along with Caltech, with Chiara Dario and Janet Roveda at U of A, and the folks at Baylor and at Missouri, and et cetera. And it's this group designed to try to maximize kind of ulcer-free days and hospital-free days and activity-rich days. If you want to kind of talk about a holy trinity of measurement items that we can do. And what's really cool is we're starting to see the rise of these, not just diagnostics, but theranostics. And it's not just heart rate, blood pressure, and glucose, but it's the smaller form factors that are really, really cool to help us manage and measure what we manage. So if we look at healing a wound, it's really kind of a complex symphony of that we just don't have time to discuss. And I'm not going to talk about an axis of evil. I'm going to talk about an axis of healing. And just very, very briefly, there are a few things that we have to do surgically and medically and mechanically to try to promote healing. And you see all of this. I'm not going to belabor all of these different technologies and therapeutics and biologics that overlap with one another. It's no different than if we were thinking of diabetes in general or cardiometabolic care. You have overlapping therapies, but wouldn't it be nice now if we had ways to measure what we're managing for each of these key areas, for debridement, for promotion of granulation tissue and regeneration, and then ultimately for looking at healing in remission for this population. And what's cool is we do now have some of these diagnostics and theranostics. So for looking at the risk for healing initially on that first aspect of things that we can look at now, bacterial load and local flow really beautifully for looking at regeneration and granulation tissue. We can now look at a whole bunch of different theranostics, and finally looking at things for healing and moving patients into diabetic foot remission. We have an even more robust kind of system. There are now a whole load of analytes that can be measured in intelligent textiles and dressings. This is just a small number of them. We don't have time to get into a lot of exquisite detail. Just suffice it to say that it is an exciting time now because we are able to, in real time, be able to identify through epidermal electronics a lot of these analytes. One of the groups in our National Science Foundation program is Rockley Photonics. I know Dave Klonoff knows these characters much better than I do, but I just met them now. And these guys now have methods, even in a watch form factor, to identify a whole bunch of analytes, not just blood glucose, not just pulse now on your watch, and even blood pressure on your watch, but now glucose and things on your watch because you have a spectrometer on there. It's just crazy. So this can do a lot of things, and we're putting this now into dressing form factors as well. Again, kind of advancing these diagnostics and therapeutics. But let's just kind of move now out of the wound healing bit in our just this sort of top level look at all of this, and talk now about healing these patients and keeping them healed in recurrence. If we can't prevent all recurrence, maybe we can prevent some in this population. And to that end, let's move forward. In the face of neuropathy, you have pressure that you can modulate externally through shoes, insoles, prostheses, and internally through surgery or physical therapy, or if you're British, or if you're trying to affect a British accent, physiotherapy. And then we can also do it through activity, by modulating activity. We haven't been able to do this in the past, but we can now because we can measure activity so well. It's really an imbalance of activity and pressure that leads to these recurrences and occurrences. And if you think about it, it's very much like an aminoglycoside or a glycopeptide antibiotic. Too high, if you want to dose your activity, and in this case, you might get an ulcer. Too low, and you don't get the cardiometabolic activity benefit. Just like otonephrotoxicity for your glycopeptide, or or lack of benefit for too low. So we can ease people in to better activity by measuring their activity. You see these data from Boston and Lieberman's lab. It's a beautiful example of a foot in motion and perfect kind of epitome of balance. The thing is, our patients are out of balance and their activity is out of balance. They often leap out of the chair, out of their sofa to go get a gift for their grandkids and take more activity in 2 hours than they usually do in 2 days. We can detect that now, and we can intervene, and we can now identify that like never before through, it's like a Holter monitor for your life, if you will. And we can now put into the first calculators that we're giving our patients, really just a high to low activity dosing and dosage, which is very, very exciting. More on that later, but variability in activity, that's what precedes a lot of bad stuff. What about, we're all familiar now with our solipsistic world of the selfie. What about now, check this out. What about foot selfies? So this is a thing now. We now have foot selfie rounds every Monday morning where our patients can just send us it's free. We just have them send us their photos of their feet, and we just collect it and put it on an Amazon cloud account, HIPAA compliant. And now we're using an AI-based Sherpa to help us identify pre-ulcerative areas along with our help. And it's amazing, and it's free. Like I said, did I mention it was free? Let me just continue moving on this area. We don't have time to go into these explainable AI models, but we're applying some of these photos that have superb predictive potential. We'll see, because again, it's garbage in, garbage out with many of these models. This could be an entire discussion, as it has been already in this meeting. Just very briefly, these wounds will heat up before they break down. We can identify pre-ulcerative areas with thermometry. And we can do this now, like you see here, with a Charcot foot in the clinic through just a simple $200 device. We can give our patients $10 thermometers and to dose this activity and identify this. But there have been entire companies now that have been working in this area to identify these wounds. And we have been working with some of them to give our patients guidelines to dose their activity by checking their skin temperature, just as they dose their insulin by checking their blood glucose. Let me fly through this because the fact of the matter is now that these thermometers can now highly accurately identify patients through intelligent bath mats, also through intelligent textiles like smart socks. And people are now subscribing to these therapies. It's exceedingly exciting. And these things now are crossing over from the most boring foot stuff into geekdom. And you see here, this is Rand Ma and others. They've won awards now at the Consumer Electronics Show, going up against virtual reality and augmented reality. Reality is winning. So this is kind of exciting. Moving those things into intelligent shoes and insoles with little shock absorbers that can identify these hotspots and get out of their own way. This is from Zoltan Pataki's lab in Geneva, or Genève, if you're a Francophile. And just finally now, we're now, these wearables are cool, but they're passé. What if we could arrange these with, just like other people that have presented at this meeting, with injectables? This is your arranged marriage. This is happening now. And you see it from Natalie Wisniewski and others, and Miguel Montero, one of our residents. They now have injectable tools that you can ping with a laser, identify all kinds of analytes, including glucose, including oxygenation. And finally, what we see here are not just injectable sensors, but in fact, intelligent conduit. This is a PTFE blood vessel. You can pair with it on the back table. And then what you can do is you can identify turbulent flow, not in the hospital, but at home. So you can stop that acute on chronic complication even as it's just starting. And this is such an exciting time to be able to be identifying this And the masters of the universe are the ones that are going to be put all this in control in some sort of graphic user interface to where we aren't having any kind of paralysis of analysis. But Madam Chair, I will quickly conclude with this. I'll show you this video. So this seems really ridiculous, right? Well, I used to play soccer as a kid and badly all the way through university. And I thought to myself, you know, when I saw this video, man, that's me looking dumb, but somehow still being in a position to help and make a difference. And when I saw this, I immediately thought of the great, not Denton Cooley in surgery, but Mason Cooley, the aphorist, who said, if you position yourself well enough, circumstances will do the rest. I believe that. And I believe we come together to try to share our successes and our failures and what's next to try to affect change. And I really think we can make a big difference. And in my little area, maybe we can keep a few more legs on a few more bodies and help our patients move through the world a little bit better, because that's what we all deserve, no matter what our health and no matter what.
Yes, Dr. Armstrong, thank you.
So I will
Dr.
stop
Armstrong.
it there. As always, you know, David, it's great to finish on his presentation, always brings a smile to the face. And has done, you know, great amount of work in this area for a long time now. We're closing up, we're a little bit over time. I did want to just very briefly touch on the activities of the academic unit and what we're also doing. And very briefly, I have a PhD student, Saskia Schwarzer, who's looking at some novel strategies for combating wound biofilms. Dr. Serena Chong's is doing some work in the gut microbiome, particularly looking at people with diabetes and who've had multiple antibiotics. We have a Masters of Research student, Taylor Phillips, who's looking at some microbial community dynamics and the lottery assembly and looking at biofilm formation in vivo. We're doing some work around validation of biomarkers in some prelim data, which we have got. And we have Dr. Michael Xi and some of the foot team, Ken Hayashi, Emma Marshall, Erica Koo, who's been contributing to a large diabetic foot infection registry we have with over 450 episodes logged, looking at clinical outcomes of DFI and the impact of COVID-19. That brings a close to today's proceedings. We've gone over time, so Professor Josephine Chow couldn't tune in to close, so I have the task of doing this, and I have some notes here, which I'm just going to quickly read through. Thanking every single person that's tuned in today, to the presenters who've put the time in to send me the presentations, be here live. A huge thanks to everyone online who's watching it in real time, and to those who are going to watch the recorded version. And this is our 4th online seminar for the Health Beyond and Research and Innovation Showcase for 2022. I think the program that we put on today just shows the high level of work that we're doing in the academic unit at Southwestern Sydney, but also the work of our collaborators in this space and area in general of diabetes, limb preservation and wound healing. Big sort of congrats here to Prof. Akinji for the grant for Acadie, which she was successful for, and myself as part of that for being the lead with Prof. Cohen and Rob Fittridge, Prof. Fittridge. Some acknowledgements to the team at the Ingham Institute for hosting the event, and for Rachel and the people in the Research Directorate for putting all of this together. Huge thank you. A thank you to the academic unit that we have with my workers, Alison France, Michael Radzita, and the rest of the crew in the lab and in the clinics that contribute largely to our work. Lastly, I would like to invite you all to tune into the next seminar in the online series, which is going to be on clinical trials, and it's going to be on the 19th of July, 2022 at 12:30 p.m. There's an exciting lineup of presentations. We have Professor Mark Parsons looking at his journey into clinical trials and research, Professor Catalaris with changing environment for immunology trials based on changes to personalized medicine, Dr. Pal, who's doing some cancer clinical trials and translational research in Southwestern Sydney, Meg Ford, who's our clinical trials manager in Southwestern Sydney Local Health District, and Miss Wilson, who's going to be looking at what the future holds for clinical trials. With that, I'd like to close. Again, thank you everyone for joining us. A few technical difficulties, a little bit over time, but hopefully you all enjoyed the afternoon, and we appreciate you joining us.